So very fitting, we should now hear from one of the Gruber Prize winners, and this is Jade Wang at Yale, and she at Baylor, and she has gotten this award for her work. Her brief bio is in your board book, and one of the things that's been very important to us is to engage these prize winners in what we're doing as well. And I have to say, I have been so pleased. Any award winner we have ever talked to, from the Gruber Prizes or others, they are always thrilled to come and talk to this group. They really have important stories to tell, and we are more than happy to give them the platforms. Jade was a physics major as an undergraduate at McGill. And then she switched to biological sciences at UCSF to study DNA replication. And this has been the trajectory of her career and her work there, and then at Baylor and now Wisconsin. So she really has gone on to do some great work, and we'll hear a little bit about this now.
I don't have
okay.
any.
Do you have enough battery or? But you have to slide it from here. Mm-hmm. You have to slide it from here.
That's okay.
Okay, so that's about one.
Wait, is it not gonna work on the?
Moving along, you have this podium mic or the one. Which one do you like?
I like to have this.
Okay.
Laser pointer. Let's search.
You might hear.
Can I have the laser pointer?
You have to slide from there.
What? The laser point? Oh, you have. Okay. All right. So it's a great honor to share the podium with Patricia Gruber and a list of distinguished male and female speakers. So I am very indebted to the generosity and vision of Patricia. And without her support, I wouldn't be standing here talking to you today. So I also like to thank the organizer for inviting me to give a talk, and also for suggesting this title, Before After the Awards, Who Notices? Let me see whether I can get the pointer. Yes. So I started my independent position as an assistant professor in Department of Molecular and Human Genetics at Baylor College of Medicine in Houston in 2006. In 2009, I was awarded the Rosalind Franklin Young Investigator Award under a search with a distinguished group of female scientists headed by Mary Claire King. So my stage is still early career to mid-career, but what I can share with you are two things. Number one, what I do, what kind of science we're working on to continue the kind of genetic research started by Rosalind Franklin. And another aspect I like to share with you is the life story that is personal so far, that is personal and is still early so far, and the memory is still very fresh of what the kind of difficulty female scientists have encountered. And Carla wants me to talk about it and really, which could offer a hint as what she wants to know, and many of you probably want to know, is that how we have so many talented female scientists who did not really rise up to a management position and didn't rise up to be the leader as they should, as they deserve to be. So Rosalind Franklin, along with Watson and Crick, show the structure of DNA. And DNA is the underlying component of the genome. And all organisms, from bacteria to human, encoded their genetic information in their genome. So the double-strand helical structure of DNA suggests how it can be replicated immediately. And this is indeed how it was done in the cell. So from works in test tube, people have figured out a long time ago how DNA is replicated, that the double-stranded DNA is unzipped. Each of the DNA template, it can then be exposed to a multi-protein complex. And this multi-protein complex, which is called the replication machine, use the single-stranded exposed DNA template to make double-stranded DNA. And this replication machine moves along the DNA template and leaving behind 2 copies of DNA, and in front of it, 1 copy of DNA. And there's an asymmetry, and that is the synthesis of leading strand and lagging strand, which is due to the asymmetry of the template itself. So we know all of these information very well. What we don't really know is what happened in the cell. Because in the cell, this DNA, which is very, very long and is in the gigantic human genome, and it's relatively small bacteria, they're all wrapped up and then packed and then put into this very limited space of the cell, of the nucleus. And in bacteria, it filled up the entire cell. And this is a bacterial cell. These are the pillars. These are the flagella. And everything inside that is blue are DNA. So how replication can really happen inside the cell? This actually remains a very elusive question for decades. The development of the genomic age really allowed us to tackle this. One thing that we can do is to sequence the entire genome and know what the sequence throughout the genome of every DNA sequence throughout the genome. And in bacteria, this complicated Raman noodle structure can be actually unwrapped using this information. And it turns out, for many bacteria, with just 1 single circle of 4 megabase per long. And the replication of the bacteria is only carried out by 2 replication machines. And they all start from this one place. One moves counterclockwise, the other moves clockwise. There are about 4,000 genes in this bacteria, and they are discrete components. And this process are called transcription that makes RNA out of it. But the replication fork is, replication machinery are moving processively along 2 replicore until they merge at the end. So this is the basic way bacteria replicate their genome. And we really need to know whether the problem they encounter throughout this replication process, because problem of replication will lead to mutation, leads to DNA breaks, that leads to cancer, human genetic disease. So to understand how this replication process going on, what we need to develop is a global genomic view of DNA replication. And that is something I've worked on ever since I was a postdoc. So what I have done is to develop a genome-wide method to measure DNA replication. What I do is to take advantage of the small genome bacteria and then use mutant, which are mutations in DNA replication machine, allowing them to perform a synchronized dance of replication. So I would have 10 to the 8 bacteria all together, and then using temperature shift along with mutant, you can actually allow them to start replication all at the same time. And this is what happened. Now you have all the replications synchronized, and every bacteria will have 1 copy of genome. And then you drop the temperature, which is what we have done. Then all of them will start replicating at the same time. What you can now look is the genomic copy number of this whole circular genome throughout from the origin of replication to terminus of replication. You spread the circle out, and then you look at the genomic information, genomic dosage. And before replication, all of the gene has the same dosage. After replication, the area near the origin of replication duplicated to be twice as much as the area further away from the origin of replication. And now they're all moving at the same time. And let's say after a while, you look at them, you will see that the replication fork has moved further away. So this is the first time we're really able to see how DNA replication is going on, not only in the test tubes on a very small piece of DNA, but at genomic level. With this, we were able to visualize many aspects of DNA replication inside the cell that we couldn't see before. And I'm just going to show you 2 slides of the kind of new regulation, new behavior of replication inside the cell that we can see. So this is a new regulation by a starvation signal that all bacteria use to sense nutritional starvation, which is called the magic spot. And then we can go for a long reason why this is called magic spot. But let me just say that these are small nucleotides that appear, magically on the chromatography people discovered 40 to 50 years ago. So this magic spot that all bacteria turn on can actually stop replication fork in the middle of, therefore constitute this regulation mechanism of cells during starvation. Because during starvation, cells don't want to duplicate their genome. Otherwise, they might run into problem and then leads to DNA damage. Okay, so this is one regulatory mechanism we have discovered that were unable to be visualized before without this type of method. And the other type of challenge that DNA replication that we review to be really a major challenge that cell has to circumvent is dealing with conflict. So again, this is the big replication machine that moves along the double-stranded DNA to produce 2 copies of the daughter strand. So in ideal situation, in many test tube situation, it encounters a very clear template. So it moves forward, and there's nothing in front of it. So if you imagine the replication machine as a car, it is really unhinged, unhindered in the test tube. But this is not really what happened in the cell. And in fact, what we found out is that the replication machine are running into a traffic jam because the same DNA is not only being duplicated to the daughter cells, but it is also transcribed into RNA. And RNA will be the important intermediate, and sometimes it itself has a function to produce proteins and enzymes and ribozymes. And ribosomes. So what this really busy activity, remember that a bacterial genome has 4,000 genes, which means there are at least 4,000 transcription units. So it would keep on running into each transcription unit. This one replication machine that duplicates the entire genome will run into multiple RNA polymerases, and sometimes array of RNA polymerases when the time of the traffic is busy. In this particular case, the busy traffic happened when cells are growing in rich nutrition media. So earlier we were talking about there's a regulation during starvation. And now the other thing we can find out is that when transcription is busy, when there's a lot of nutrition, there's a different challenge for the replication machine. And this is what this creates is a head-on collision between the replication machine and transcription machine. And what we now know is that this itself can create DNA damage, which leads to disease. So this is just a visualization using a similar method I've shown you earlier of replication running into a busy area of transcription, running into this traffic during growth in rich medium. And during growth in poor medium, there is much less transcription traffic, and replication is moving without that much block. So you can really see stuff happening throughout the genome. And this is a microarray profile that has the entire bacterial genome. And this really reveals where the replication ran into problem, where it has less problem, under what condition it has problem, under what condition it has less problem. Okay, so what is the implication to health? So what I want to remind you that what we're doing is a very basic research. So we're not going to see drug development unlikely in the next 10 years with the kind of basic knowledge we gain. But what we can really learn is fundamental new principles that can be expanded to higher organism, to more medically related research. So for example, what we can learn from our little story with bacteria. So what we know is that human genome is highly susceptible to damage. And when many of this DNA damage will lead to genetic disease, aging, and cancer, right? So we know of many causes that are well known, such as ultraviolet radiation, and we put sunblocks on our face. And from yesterday's speaker, now we know that sunblocks themselves are very dangerous because they're nanoparticles. And then we stop smoking tobacco because we know tobacco can damage genome as well, right? But the story we learned from bacteria is that the problem of the duplication process, the intrinsic DNA replication process, is also a source of DNA damage. And in addition, nutrition, such as amino acid availability, which was thought not to be directly interacting with the genome, can, by affecting DNA replication, causes DNA damage. And that could be a potential source of genetic disease and cancer. And this could also tell us that the nutrition we take as human might actually indirectly affect our genome and mutation. And that when we're thinking, when we're taking diet, it is not just taking a lot of orange juice and don't take McDonald's fast food. That might be bad for us. And sometimes we want to think about many of these processes are very complicated and indirect. And that is the story we learn from bacteria that can guide us through the understanding of humans. What's more important is that many of the processes involved in this regulation are encoded by genes that are conserved from bacteria to human. And some of these genes might have functions in protecting as both oncogenes and tumor suppressors via their ability to influence DNA replication, via their ability to introduce DNA damage. So this is one thing that we've learned. In the sense of basic biology, it tells us that the central dogma of molecular biology, such as replication, transcription, and translation, not only should be studied independently, as we learn from the biology textbooks, but their interface is also really important and has a fundamental impact in the cell. And in another way, let me just summarize this, is that these processes have much more extensive interface with each other and with metabolism. And any type of stress, including nutritional stress, would interfere not only the process, the central dogma process, independently, but it would interfere their interaction with each other. So what we need is a complex global view of how cells control multiple aspects of their metabolism. And this is what my lab now has been taking on the new direction after the Rosalind Franklin Award, which is to combine multiple approaches, because we really cannot just use genetics or biochemistry or genomics to understand some of this question. What we need is a combinational, integrative approach. And the microorganism, bacteria that grow very fast and can easily to manipulate genetically, can provide a good platform. And for example, here is one of the flowchart of one of our papers that we recently just published. And in this paper, we use the combination of metabolic profiling, which is liquid chromatography, mass and tandem mass spec, with microarray-based transcription profiling, with genomic whole genome sequencing, next generation whole genome sequencing, with computational approaches. And then from prediction of enzymatic target, we would actually go into the test tubes and look at enzyme kinetics. And on parallel to it, we'll perform unbiased genetic selection. So here, the high throughput approach, including genomics, transcriptomics, and metabolomics, are combined with genetics and biochemistry to review new fundamental aspects of homeostatic control and viability, cell viability, which has implications so far we found in antibiotic resistance of microbial, as well as potential implications in human disease mechanism. So how do I get here? And so this is the second part of story. And this is integrated with the first part of story in the sense that many of what I showed you earlier scientifically involve a combination of somewhat of a physics approach and computational approach with biology approach. And that stems from my training as a physics major. And even before that, when I was brought up in China in high school and in elementary school, I was heavily involved in math and physics. And China has a very rich and history of scholastic competition, and scholastic achievement is highly appreciated. So basically, my life was involved in many aspects of competing for scholastic awards, which I'll talk about later. And so this is the high school of Peking University, which I went to. And then at the end of high school, I had this high school years where I was in this special class for physics students and the physics high school students. So there are 26 students in my class, and me and another girl are the two women. And then we stay in schools, and we're roommates, and then all the boys, they're in different rooms, they discuss science, and we sort of occasionally do it. And then very quickly, I was in one of these special training class for this International Physics Olympiad. And this time, we had another 22 people, and I was the only woman. So, and all my teachers are guys. So it is, in a way, very isolating. But at the time, I did not know that was the environment, right? I just felt, maybe I'm not really competent, because that when the professors are teaching classes, they don't really look at me. They would go and then they would talk to a guy, and they were they nod and move on to the next slide. So I had this kind of isolation, which at the time I did not really know was an issue of the society. I thought it was an issue of me. So the first time I really get to know women, and I think really I get to know a group of excellent women, is that after I went to college, we went at Peking University as a freshman year. I went through this forced military training. And in this military training, all the freshman year went to this military school, and I roomed with other excellent female undergraduate students, freshmen. And everyone in my dorm are biologists. And what they told me is that their parents told them that the 21st century is the century of biology. So this is a little, and this stayed with me. I really enjoyed my 1 year in military training, unlike most other people. So even though we have physical activities, but on the other hand, to talk with this brilliant group of young women was fantastic. It's intellectually really stimulating. I went back to physics. In fact, I went to physics because I've always think that physics is my calling. I was good at it, and I was interested. So I went to McGill University in Canada, and this is the Rutherford Physics Building. McGill University's former chair was Rutherford, who won the Nobel Prize for his contribution in atomic physics. And this, you can see, is a concrete building that has a large basement where the undergraduate students hang out, and all of them are guys. And all my professors are guys. And then in the summer, I get to go to National Research Council of Canada to do summer research. And this is one really nice thing about the Canadian system, which unfortunately they discontinued, is this Women in Engineering and Science program, where National Research Council of Canada will select about 15 women every year. And we would be committed to do 3 years of summer research, 16 weeks every year. And then we will be given more than $10,000 every year for that. So basically, that financially provided my family, who was a new immigrant with very limited financial means, and also as well as provide me with all the opportunity to do summer research every year. So I was studying nanostructure quantum dots, and I've had a lot of fun with it, and we published. But when I was moving for graduate school, this intellectual, exciting conversation I had with my roommates in military school comes back, and then I decided to check out biology. So I interviewed a place like UCSF, and this is UCSF with Golden Gate Bridge. I fall in love with the city. Most of all, I fall in love with the science people are doing. And one conversation I had that was really very impressed with is, of course, with Cynthia Kenyon, who studies aging in C. elegans. So I went to biology, basically. And this really changed me because at first I was making this difficult transition into biology from physics. And I didn't even know you can use the peer group of women because this wasn't really part of the culture as a physics student. But gradually I developed this and I really get to know a lot of female mentors. And one of them is Carol Gross, who has been really important for me. And she was actually the first person who said, "You're smart." So that was such a reassurance because I've never heard of this despite being winning national first award prize for physics competition. Nobody said I was smart. I always thought one day the boys are going to catch on because that's what people told you, right? When you're straight A, they said, well, they haven't really figured it out. They're going to figure out. When they figure it out, they're going to perform better than you are. So that's sort of the idea. That's an event that I've been waiting for all my life, that the boys to catch on. And it hasn't really happened, fortunately. And I don't think it's going. So here I studied chaperone-assisted protein folding with Professor Jonathan Weissman. And then also helped by the mentorship of Carol Gross. So then I went to Boston. So I went from West Coast to East Coast at MIT under the supervision of Professor Alan Grossman. Here I fall in love with microbes, small bugs. And this is the first time we start to think about using genomics to study DNA replication. So Jonathan taught me biochemistry. And before that, I learned about physics. Now I learned genomics. And then really it is at Baylor College of Medicine when I started my tenure track position in molecular and human genetics that I learned genetics. And I learned genetics from a senior colleague of mine, Susan Rosenberg, who is also an excellent microbial scientist. So not only about that, but we would share stories about how she and I feel in a lack of confidence as a female student. And she said that's actually called imposter syndrome, Jade, it's called. And then another person who really helped me is Huda Zoghbi, who is the Gruber neuroscience prize winner. And she is one of the most fantastic colleagues I've had. And when I would have problem supervising my students, because I don't really want to be there's this common stigma that female scientists are really tough. And then I wasn't really sure whether I should be tough or nurturing under certain situation when my students are having issue. So I would talk to her and I would be saying, well, I don't want to say this because I would be considered too tough. And she would tell me that, no, your issue is not being too tough. You need to be firm. So you're too sweet if you can't do this. Sometimes you have to be firm. And then using her strategy, it would actually solve the problem with student mentoring immediately. So the other group, other than female role models and female mentors, are female students. They're the first group of people who trusted me and joined my lab. In the first 3 years when I started at Baylor, rotation students are all female. And then the female student eventually joined my lab. And they really built the success of my lab. And in 2009, we had our first male students. And it quickly balanced. So the way that I built my lab, which I find very helpful, is to introduce diversity into the lab. And I think both gender, racial, and cultural diversity is actually very helpful for people to really learn how to work with each other. And one issue I really had is to work with my Asian students because I understand them and understand their issue. And so I really want to work with them so that they can intermingle. They can really become part of the teamwork. So, gradually, interacting with all the female mentor and role models also make me start to look beyond what I've done, which is research, and into a more organization and service role. And for example, I don't know how many of you have watched. Yes. So this is, So yes. So this is the first year I took on organizer of Baylor College of Medicine genetics retreat. So we would have this entry of skit of video. And this one is called Lady Scientist, or you can Lady Gaga Baylor College of Medicine. You can probably still find it on YouTube. And it was so fantastic. And these students are using, these are Eppendorfs. I mean, these are pipette, these are experimental, and these are garbage, biosafety garbage bags. So what they do is they make this dress and they played it, and everyone's had a lot of fun with it. And it was uploaded on the internet, and this group won $1,000 for making this skit. But in the end, when they would load it onto the internet, I think it's 2 weeks, have 2 million hits. So this is one of the most viral most viral skit. So this process really expanded my horizon, and I just finally like to talk about the Rosalind Franklin Award experience. And you've heard most of it through this history. But I want to say, and in particular in referring to what Patricia have talked about, which is the nomination process. So before the nomination process, before the application, this one I think is self-nominated. I didn't think I have a chance. Like many women, we don't think what we do is important and interesting. So my senior colleague, Susan, really said, you can have this chance, Jade. You are from physics. You work on DNA, and you're trying to look at genetics from a different perspective, from a new technology. So under her prolonged persuasion, I decided to apply. And then my female colleagues are the people who wrote me the letters. And they got me the award. And this really shows, and so far I find that many of the promotional force in my career are women or men who really understand that and support female scientists. And after award, one major thing that is really, other than helping me with publication, visibility, getting tenure quicker than what's in the tenure track, what I think is it expanded, because it increases confidence, it also expanded the research horizon and into what I truly interested, no matter what kind of technology, we feel brave enough to move forward towards it. And the other aspect is the expansion beyond research. So I think this award, which is awarded to an early career scientist, really provide us with more opportunity than just the money visibility. For example, what I find really helpful is to be invited to go to the Gruber Award prize reception for example, the Genetics Award, and where you really get to meet, and I get to meet Mary Claire King, I get to meet many fantastic people, which really make me start to think about that what we should do is we owe the society, as those who were promoted by female, we owe the society to really contribute back. And another aspect is by contributing back, it expanded the horizon of our vision. And being on Rosalind Franklin Society Advisory Board, I was invited after winning the Rosalind Franklin Award. I went to earlier board meetings, and it's just so educational, and to know what's really happening in the world. And the one person, Alice Huang, whose talk I gave previously, really set, in a way, a role model of what I think a female scientist from a Chinese or Asian heritage could have. And one aspect is this issue about bamboo seeding, which I've never thought about, that glass seeding not only exists for female, but it also exists for Asian. But that transition is from this kind of research position to managerial position. And what we can do as Asian, how do we change our culture? And how do we change the perception of the society to Asian scientists? And is actually a really interesting issue that I get interested. So in a way, this award really is a word I know that no boys are going to catch on. And the issue is no longer self-discovery, self-confidence, and confirmation. It is more about making the impact in the society, doing exactly the kind of science we want to do, and then basically have fun with it. So I've recently moved to University of Wisconsin-Madison. I mean, still in the transition. And this is where Joe Handelsman was the department chair of bacteriology for quite a while. And I moved into the same department. I talked to Joe, and she reassured me that just like Baylor College Medicine, which is an extremely collegial place with support for female faculty. And this University of Wisconsin-Madison is the same type of place. What they offer in addition is this rich microbial community, one of the best microbial community in the world, as well as the opportunity to teach undergraduate students, which I think is a really, from my own experience as undergraduate student, I think it's a really important process to build their confidence. It didn't happen to me, and I sometimes wonder what would have happened if I had stayed in physics, because I might not be mentally prepared for the difficulty. And as we know that extremely successful people who really know what they want to do, they might exist, and a lot of them is why they're successful. But then there are us who are sort of there's a stochastic chance that we're successful, and there's a stochastic chance we're not, because we're still learning. And our scientific journey is also our personal journey. And for those of us, I really think we need help. And I have gotten my help. I'm very, very fortunate. In many sense, looking back at physics, especially going back to some of the physics department, I really noticed that we are so fortunate that we have female mentors, we have community of support, and this actually is very important. As what we want to promote is not just like physics where you have the 5% quota of female faculty. What you need is really 50%. And then that's really a threshold. And finally, just like to conclude the transition of the Rosalind Franklin Award to now. And the last thing I did recently was on the Franklin Rosalind Award review committee with Mary Claire King and other scientists. And it's a fantastic experience. We had two people we really want to be given to award for the model organism, which is the award I have. We could only give one, and we give the other one honorable mention and encouragement. So I think there are many of us, and please, if you have any questions.
We'll have time for discussion afterward also, as it sets in.
Yes.
Since I'm in physics, I greatly enjoyed your talk.
Thank you.
What are you doing with the physics people in Wisconsin? There aren't too many of them there, but there's some. I gave a talk there recently.
Mm-hmm.
I don't know, maybe before you came. Very recently was there. Mm-hmm. They don't have very many women there, but what are you doing to promote that?
Yeah, so there was this yesterday, one of the speakers talked about MDs, right? It takes 30 to 40 years. Basically, it's one generation, or maybe more than one generation, in terms of reproductive cycle, not just in terms of undergraduate generation, which is every 4 years, to really increase that number. So I actually did get a physics offer from one of very good theoretical and math department for job offer assistant professor. But during the interview process, I would run into a certain situation that I get used to because I was an undergraduate physics major. And then there was one person on that search board who is a female, and she's from biology. She came to me afterward and I said, I cannot believe how this person treated you. It's so chauvinist. And then I was like, oh, yes, that actually does exist in physics. So basically, I was asked a question that is basically a qualified level question during my job talk. I answered it and said, is this a qualified question? And he just stood up and left, right? So this was the circumstance that happened. So I think it's one of these situations where you wouldn't think is discrimination against women if you were a student, because you get used to that. But I think in a way, maybe it reflects certain aspects. And again, I didn't really feel bitter or angry about it, but it did make me to think that maybe just being a female undergraduate student, I get used to certain way that we are sort of slightly belittled. And then we form self-evaluation because we doubt ourselves rather than how we're treated first. And so in this particular situation, it really makes me feel maybe there's really a big culture rift. I don't know how to help the situation with women, but I think really what we need is to increase the female promotion of that quota of 5% towards 10%, towards 20%, and it probably takes generations to build it up.